Good afternoon, everyone, and thank you for joining us this Friday lunchtime. My name is Elizabeth Mamali, and I'm the Postgraduate Researcher Development Officer for the Bristol Doctoral College at the University of Bristol. Welcome to the semi-finals of this year's virtual 3-minute thesis competition. For those of you unfamiliar with 3-minute thesis, the competition challenges doctoral candidates to present a compelling spoken presentation on the research topic and its significance in just 3 minutes. Traditionally, our participants present in front of a judging panel and live public audience. However, due to current public health guidance, we have had to take a slightly different approach this year. Our semi-finalists have already delivered their presentations to a judging panel, and they are eagerly awaiting the outcome, which will be announced by the chair, Professor Harry Meller, at the end of today's broadcast. 6 of today's competitors will then have the opportunity to take part in the university's final next week, which we hope you will join us in attending and use your opportunity to vote in the People's Choice category. Thank you again for joining us this afternoon, and best of luck to all of our competitors.
According to the number of deaths, according to COVID-19 infection until today is more than 300,000 cases. The number of deaths according to SARS-1 coronaviruses and MERS coronaviruses infection until today is more than 1,500 cases. Since 2002, these coronaviruses start to infect humans, and it stops our life these days. Since 2002, there is a real war behind the normal life picture between the scientist and the coronavirus family. To solve the coronavirus's mystery, we need to study the reaction between these viruses and the human cell on a protein level. Why it's protein? Because the protein is a building block for all organisms, include human, animal, and even viruses. Can you imagine that these viruses are not able to produce a normal, an essential protein for them? They need a living things to live. They need a host protein. My project is doing an experiment from 2 phases, as it's shown in my slide. The first phase is we're gonna infect a normal cell with these coronaviruses in the lab. After 24 hours, the coronavirus protein should react with the human protein. Then we're gonna harvest these cells, put it in a sample tube, analyze this sample to define what is the human protein that have been produced from these cells after it has been infected. Let's say this AVDAC, we're gonna use AVDAC1 as an example, as an essential protein. In a second phase of my experiment, as it's shown in my slide, we're gonna genetically modify a human cell are not able to produce AVDAC1. And then we're gonna infect this cell with the coronaviruses. If this cell are not able to be infected with the coronaviruses, that means that AVDAC1 is one of the essential human protein for these coronaviruses. In that case, our scientists will take their sword like a transformer and cut this protein-protein interaction between a coronaviruses and human cell by using a certain kind of medicine or treatments. If we find an essential human protein for these coronaviruses, I mean SARS-1 coronavirus, MERS coronavirus, and COVID-19, we're gonna beat this family, even if they have a 4th one. Thank you so much.
Imagine that you are in a coffee shop drinking coffee and you want to know what happened with the grains inside your mug. Probably they will be thrown away. On the other hand, you're having a conversation with your friends and you want to go to Australia to go scuba diving because you love fish. But at the same time, you are concerned about global warming and you know taking an airplane means greenhouse gas emissions. What if I tell you that airplanes could save fuel consumption by mimicking your favorite fish and reducing the coffee grains? Avian wings like hawks and eagles can fly silent and efficiently because they have flexible wings that allow them to change the wind shape in a smooth and continuous way. That will keep the airflow attached and therefore reduce drag and noise. In contrast, aircraft wings are very stiff. They cannot change the wind shape. That's why they require control surface like ailerons that aggressively separates the wind flow from the wind surface, leading to a substantial increase in drag, noise, and fuel consumption. On the other hand, I am working with a flexible wing inspired by the fish skeleton. It can change the wind shape in a smooth and continuous way to keep the optimal shape and the wind flow attached, exactly like avian wings. That's why it can reduce the drag, noise, and the fuel consumption as well. However, flexible wings lack something. They are too floppy to hold and lock the optimal shape. In other words, I got the skeleton to change shape, but I'm missing the muscles to lock and hold the shape. So how do I get the muscles? Well, have you ever held a vacuum packed ground coffee? It looks like and feels like a brick. Try to bend it and you will realize that it's so strong. But why it's so strong? Well, it is the vacuum. The vacuum squeezes and locks the grains together, make it into a solid brick. But if you could open the bag, the vacuum is released, the grains are loose, and the whole structure behaves like a fluid. We can mimic soft and stiff muscles by using vacuum packed particles. My flexible wing has empty chambers along the spine. If I fill it with coffee grains, I have a new concept that can switch the stiffness in the spine by playing with the vacuum. So when the vacuum is off, I can change the wing shape. And when the vacuum is on, it can lock and hold the shape. So now I have a smart wing that can switch the stiffness in order to save fuel consumption in airplanes. Now, you will be guessing why coffee grains. It's not because they are tasty, it's because their shape is rough. That roughness increases the friction between particles and therefore create an interlocking effect that increases stiffness. So the next time you drink your coffee, you will know how to make greener airplanes. Thank you very much and enjoy your coffee.
How do we make sure that we're in the right place at the right time? Can you cast your minds back to our world before lockdown? Imagine our cities like on the top left of this slide, constantly in motion. Traffic moving on roads to get everyone and everything where it needs to be. Like the camper van on the top right, taking surfboards to the beach. Pioneering engineering efforts have led to complex transport networks. But I've got news for you. Evolution did it first. Inside every cell in our body, transport processes are essential to stay healthy. And whilst we might be stuck at home, inside our cells, transport goes on. As you're sat here imagining driving down the road, right now, billions of nerve cells in your brain are working hard to communicate through electrical signals in an intricate neural network. To send these messages, signaling cargo must be trafficked long distances through each nerve cell and to the next. Transport inside cells in the brain is essential to allow us to see, hear, think, feel, and move. The roads of our cells are a network of filaments called microtubules, shown on the bottom left of this slide. These assemble throughout the cell and form the tracks for transport, our cellular motorways. The cars are motor proteins, shown in red on the bottom right. These pick up their cargo and walk along the tracks to deliver them to their destination. They have motor domains which act like feet and allow them to literally walk step by step along the microtubule tracks, dragging their cargo with them. If you can escape from your fantasies of unrestricted travel for just a second, you might start to remember that sometimes transport goes wrong. How many times have you been stuck in traffic due to an accident or roadworks, or delayed by cancelled bus or train or plane? Sometimes things go wrong inside the cell too. When this happens in cells in the brain, things end up in the wrong places, and eventually that can lead to neurodegenerative diseases, like Alzheimer's or Parkinson's disease. I'm researching one of these motor proteins to find out how it works, and in particular, how it picks up its cargo. Ultimately, if we can understand the mechanisms by which these proteins work and go wrong, that will help us to understand and treat brain diseases. I've designed a new tool which allows cargo to attach more tightly to this motor protein, increasing the amount it's transported. The position of different components in the cell depends on a delicate balance between motor proteins acting in different directions, a bit like a tug of war. And much like a really muscular teammate, our tag allows us to steer transport in one particular direction, giving us control over the position of components in the cell. I'm dreaming of a world where we can understand and control transport in the cell to generate a biological breakdown recovery service and prevent these debilitating diseases. There's still a lot of work to be done to understand the amazingly complex transport networks that biology has engineered inside our cells and how these break down. But by developing tools to take control over motor proteins, we're taking steps to get things moving in the right direction.
How do we take a pile of stuff and make it into a material? Really, the only difference lays in the function that we impose on that material. A paracetamol tablet is just a pile of atoms until we realize that ingesting it may take away the headache that I have from one too many glasses of isolation wine. My PhD looks at the ideas of material science. How do we take the building blocks that make up the world around us and put them together in such a way that we can change some aspect of our lives? To me, this process is split into 3 sections. Number 1 is desire. What do I want? What would be the perfect material to carry out the process that I want to carry out? Number 2 is then a bit of forethought. To create these properties, what will my final material have to look like? And then finally, we have construction. Knowing how the building blocks should be put together, how on earth do we get them together in the correct fashion? In the pursuit of such a process, my research looks at putting together some of the smallest building blocks in nature in an ordered and often beautiful fashion in the form of crystals. Spring to mind. But more importantly, most of the medicines and pharmaceuticals that we come into contact to on the day-to-day also come in the form of crystals. A critical part of making pharmaceuticals that can have any effect on the human body is making sure the building blocks are put together in the correct way. A crystal is a material made of a single building block, put together in a repeating pattern. A simple way of viewing this is like a well-built brick wall. As we're putting together our crystal, we realize there's not just a single way of putting together the building blocks. Even using our analogy of the brick wall, we know that we can stack the bricks on top of each other in different ways. The ability for a crystal to form in different structures is known as polymorphism, meaning many forms. My research looked at changing the conditions under which crystals grow to try and persuade different polymorphs to form. When I'm successful, I can see the dramatic changes that changing the structure of material can have. The yellow and orange crystals shown on the slide are made of exactly the same building block, but they're just stacked together in different ways. This is a very visual way of seeing how materials, the structure of material can change their properties, but this relates directly to pharmaceuticals. The way that the pharmaceuticals are put together inside of the drugs that you take dictate whether they pass straight through your body without having any effect, or whether they're going to make you better and be absorbed into the body. And these critical reasons are why material scientists like me are making new tools to alter the structure of crystals to make better pharmaceuticals for our future.
Have you ever donated blood? From treating long-term patients with blood-based diseases to providing their life-saving needs in emergency cases, blood transfusions, made possible by your donations, have become a vital component to modern medicine. In the UK alone, 2 million pints of blood a year are required to fulfill the medical needs of our society. To put this into context, as a nation, we consume almost 4,000 times this in pints of beer. However, unlike the brewing industry, we just simply don't collect enough blood to meet the demands of our ever-growing and aging population. So over the past decade, scientific research has instead begun to find ways in which we can grow red blood cells in the lab. This would mean that A, we wouldn't have to solely rely on your blood donations, and B, we wouldn't have to worry about screening that blood for infectious contamination. Incredibly, we're now at the stage where we can produce red blood cells outside of the body, through mimicking natural processes. What starts as an unlimited pool of early blank cells, called stem cells, in a glass dish, we're able to nurture and gently encourage the formation of fully functioning, mature red blood cells. Now, this is impressive stuff, but I want to go one step further. I want to be able to produce blood that not only functions to provide our bodies with the oxygen it needs, but that simultaneously functions to help us fight disease. I'm exploring how these cells can be modified to help neutralize the damage caused to tissues during organ transplantation, wound formation, and cardiovascular disease. To do this, I'm using pioneering techniques that triggers the stem cell's DNA to produce new and helpful disease-fighting cellular machinery. Now, once these cells have fully matured, they'll naturally contain zero DNA. So this provides us with a long-lasting therapy that poses no risk of causing DNA changes anywhere else in our bodies. So perhaps akin to that super serum that made Captain America, our enhanced red blood cells will also help to keep some bad guys at bay. I'm still in the process of trialing exactly which cellular machinery and in what combination is our best line of defense. So I'd hold on to your tablets for now, but look to a future of Blood 2.0, lab-grown blood that combats disease as well as delivering us oxygen.
When conducting medical research, one might focus on stopping a disease that could end a life. What about a disease that doesn't just threaten ending a life, but could stop and prevent a child from starting one to begin with? Congenital heart disease is known to be the highest recurring form of pediatric heart defects, and it occurs in 75 out of every 1,000 live births. These children struggle to survive given the current repair methods. The prosthetic replacements used today fail to grow along the child's heart as it grows and develops, and therefore the child would need several open heart surgeries throughout their life. And with every surgery, increases the risk of death and reduces the quality of life. My project's main purpose is to develop a natural material that eliminates this multi-stage repair process. I'm using the amniotic membrane from the mother's placenta to fabricate a biocompatible material that is flexible enough to grow with the child and at the same time still has the strength to support the beating heart. This material would also be seeded with mesenchymal stem cells that aim to encourage the heart to regenerate and repair its own defected parts. This way, we would be able to treat the child or repair the heart in one step instead of the multi-step process, and this would increase the quality of life and reduce the death rates. So I've successfully developed the amnion-based scaffold, and it has been optimally processed in both single and multi-layers, depending on where in the heart it would be needed. And I've tested it in vivo using piglet animal models, where it's shown that it was able to adapt in the natural environment of the heart. And the biomechanical characteristics tested showed that my material is flexible and as strong as the native heart tissue of the animal. And when I came to seed my material with stem cells, the cells were happy to inhabit, grow, and survive on my amnion. And this would indicate that there is room for it to interact with native tissue, signal native cells, and encourage the heart to regenerate and reconstruct its own defected parts. And since the material comes from the placenta that the baby grew in, this would minimize the risks of infection and rejection during transplantation. And what better gift could a mother give to its child? Thank you.
The Nobel Prize-winning physicist Richard Feynman once said, "What I cannot create, I do not understand." This is true of many niche areas of science, not least my own. But I really quite like this quote, and I'll tell you why. I'm a protein designer by trade. Mother Nature was also a protein designer. In fact, she was the pioneer of the field. I'm very much in awe of what nature has done, but to be honest, I don't understand it. I don't understand it because I didn't do it myself. Proteins carry out processes that are essential to life, like carrying oxygen around your body and helping you to digest your food and fight off nasty infections. Proteins are built from a Lego set of 20 amino acids, like those you see on the top left of the slide. These amino acids are linked together to form long chains, which then fold up into 3D shapes, like you see on the top right. However, we don't fully understand how we get from amino acid chain to protein shape, and this is called the protein folding problem. In my PhD, I'm trying to solve a smaller version of this problem. I want to build a new protein. It has similar features to natural proteins, but it's an entirely new shape. It has never existed before. I have the Lego bricks, and I can look at some examples that nature made earlier for inspiration. But ultimately, there is no instruction manual or picture on the box that shows you how to build something brand new. On the bottom of the slide is a picture of the bullet cluster. Here, NASA found matter, shown in red, as well as dark matter, which is coloured in blue. Now, I guess you're wondering, what has space got to do with protein? Well, the matter reminds me of natural proteins, and the dark matter makes me think of all the proteins that could be. You see, we can explore proteins in the same way that we can explore space. We'll never know what's out there until we go and look, and there are a lot of dark matter proteins to explore. Why let nature be the limit? We could build new materials, new therapeutics, and we can understand even more about our own biology in doing so. So what I like so much about that Richard Feynman quote that I mentioned at the start is the truth it holds. What I cannot create, I do not understand. It's true, and so is its inverse. What I can create, and I will understand.
Somewhere in the middle of the English countryside is a research facility, where scientists are trying to produce clean, practically unlimited energy from a miniature sun. In these experiments, scientists fuse or stick together molecules, and this releases energy, just like in the sun. One of these molecules, called tritium, is radioactive. Now, don't panic. You'll get about as much radiation from the tritium used in these experiments as you would from eating a banana, but that doesn't mean that we don't want to know how much of it there is and where it is. So my research focuses on designing a radiation detector for the tritium that we can put into a machine producing as much heat and pressure as the sun, and over many years will be able to withstand the radiation equivalent of thousands of concentrated bananas. Turns out the ideal material for this use is diamond. We think of diamond as big, blingy jewellery, but diamond has a myriad of fascinating electronic properties which make it ideal for use in devices such as radiation detectors. We can now grow diamond in the lab, so we can grow it to the exact dimensions that we would need for use in a radiation detector like mine. So how does this work? When tritium undergoes radioactive decay, that is, it loses the unstable bit that makes it radioactive, it releases an electron. The more tritium there is, the more electrons there are, these red wavy lines on my slide. My detector is made up of a thin diamond film with a layer of metal on either side, which makes it part of an electronic circuit. And in this circuit, it measures the number of electrons that are present that are hitting the detector. By measuring these electrons, we know how much tritium there is because the tritium is giving them off, so we know how much radiation is present. Similarly, if there's no electrons, we won't be making any readings, so we know there's no radiation and no tritium present. Now, all this sounds fairly simple. Take a piece of diamond, stick some electronics on it, and shove it down there in the corner near the mini-sun, and we can measure how much tritium there is. Simple, right? Though, if it was, this probably wouldn't be the topic of my PhD. There's plenty we don't know yet. We don't know how thin the diamond needs to be. We don't want it to break, but we need the electrons to become part of the circuit so they get measured. We don't know what other molecules might be present that could also be releasing electrons and telling us there's radiation when there isn't. And we don't know what metals and materials will be needed to make up the electronic circuit that can also withstand the same conditions that diamond can. But maybe, once I've worked all this out, I'll be able to tell you how many bananas worth of radiation is near a miniature sun somewhere in Oxfordshire. Thank you.
Last year, I was cycling home late at night when a badger ran out in front of me. It went straight underneath my front wheel, sending me flying over the handlebars. Fortunately, both myself and the badger were absolutely fine, but I did end up with deep cuts across my knuckles. And the first type of immune cell to be recruited to these cuts would be neutrophils. And my research focuses upon this essential cell type. Neutrophils are visible to most of us as the main components of pus. However, they are far, far more important than that. They act as the body's primary defense against invading pathogens. Neutrophils circulate in the bloodstream and are rapidly recruited to sites of infection and also tissue damage. Upon reaching sites of infection, they then clear invading pathogens through numerous different mechanisms, which I've actually highlighted here on the left of the slide. They engulf bacteria directly in a process known as phagocytosis. They also release proteins from granules, which actually target pathogens. And they also shoot out their DNA to form these nets which entangle bacteria. And neutrophils do all these things despite having an extremely short lifespan of only approximately 8 hours. And what this means is that our body has to produce neutrophils at a simply phenomenal rate. Neutrophils are produced from stem cells which reside in the bone marrow. And in the time I've been talking, you will have each hopefully produced around 50 million neutrophils. And I say hopefully, because when neutrophil levels drop, we are in serious trouble. And this is especially clear in the case of cancer patients. So when cancer patients are given chemotherapy treatment, neutrophil production is disrupted. And without neutrophils circulating in these patients' bloodstreams, they are prone to potentially life-threatening infections. And currently, a skirmish with a badger would have a significantly worse outcome. And currently, such patients are treated through neutrophil transfusions. However, this is rather inefficient as most of the neutrophils are actually in the process of dying. To address this, what my research does, I'm aiming to produce neutrophils in the lab. I take blood left over from blood donations and isolate stem cells from these samples. I then expose these stem cells to numerous factors within the bone marrow, which normally reside within the bone marrow, forcing these stem cells to actually mature into neutrophils. And I'm currently using this as a model system to examine diseases in which neutrophil production is disrupted, a process that normally occurs within the bone marrow, so it's very difficult to study. Importantly, I've shown that actually these lab-grown neutrophils can possess all the bacterial-killing mechanisms I mentioned earlier, which means long-term, if we scale up this culturing protocol, we can potentially use it therapeutically as a source of fresh patient-matched neutrophils for cancer patients, as an alternative to the inefficient transfusions. Thank you for listening.
I can report back that we've had a selection of fantastic talks this afternoon. It's been a real pleasure to be reminded at this time of just how great and varied the research is that's carried out by postgraduate students in this university. We have some semi-finalists. It was incredibly difficult for the judges to separate these talks. All of the talks were excellent. There were no bad talks, but we do have to narrow it down to 6 students, and in no order those students are Hannah Langlands, Debbie Nicol, Jessica Cross, David Brigido, Sophie Osbourne, and Ted Roberts. And so congratulations to those semi-finalists, commiserations to the runners-up. As I said, everybody was excellent, and we'll look forward to seeing those presentations in the final.
Thanks, Harry, for your verdict, and thanks everyone for attending today. Remember to visit the University of Bristol Facebook page at 12:30 p.m. next Friday to watch the fast-paced final and help choose this year's People's Choice presenter.